Okay, so we're going to finish up chapter 26 here, and we've kind of covered already most of or a lot of what's happening, about half, I don't know, whatever. Um, but we've covered some of what's going on with the first part of the nephron, where you have the afferent arteriole where blood moves in, and then it's filtered. So saw that we have filtration that happens here at the glomerulus. So some stuff is filtered, and the fluid will move in with the ions and other things like glucose will move into the nephron, and then some of it, of course, just goes right on past. Um, but about 20% of it moves into this, and then we see that most reabsorption takes place in this proximal tubule. Okay, so that means that as soon as these particles that were formerly in the blood are filtered through and go into the nephron, most of them are reabsorbed right away in the proximal tubule. So things like sodium are brought back in, and we've discussed how that takes place. That sodium is actively pumped out of the cell, the sodium-potassium pump. It's everywhere. And then that sodium will draw other things in, like here's chloride that's being pulled in, glucose is being pulled in with the glucose transporter, amino acids are pulled in, and then when all those particles are taken out of, so this is inside the nephron. So as these particles are moving out of the nephron, then water will follow that through osmosis. So water is going to follow those particles, and then water is reabsorbed as well. So that's what keeps that stuff in the body. And then we talked about how it changes in the descending loop of Henle. In the descending loop of Henle, we see that it's permeable to water, but not salts. Okay, so now we have some selectivity. And in the ascending loop of Henle, it's permeable to salts, but not water. Okay, and so we have those differences. And ordinarily, we would see that if this were just all by itself, um, doing this all by itself, that we would see that it's very dilute on the left, and then it's very concentrated on the right. But that's not the way it is. And there's a reason for that, and that's the countercurrent mechanism. So as this water, so here we go again, we can see the water moving out. So as the water moves out, and from the descending loop of Henle, it's immediately swept up by this thing called the vasorecta. Okay, the vasorecta, it's just a capillary, but this vasorecta is flowing in the opposite direction. And so as soon as that water moves out of the nephron, it moves into the capillary and is pulled up to the top, which is going to make this top part more dilute. Okay, so you might say, okay, well, what's drawing that water in? Why does it want to go in there? Water moves from a low concentration to a high concentration. Well, it's because of what's happening on the other side. All of these salts that are moving out of the nephron over here on the ascending limb are also pulled into this capillary and are swept down. Okay, because if you look at it, it's dilute up here, the water is being pulled up, and of course this is a cycle, so it's happening all the time. It says, wow, look how dilute it is up here, and so I'm just going to move up into this area, but then immediately it's pulled down because that's the direction of flow. So that continues to happen, and then the concentration gets a little bit higher, but the concentration is also higher down here. And so that builds up the concentration down deep, and so we end up with a gradient where we have it's very dilute, which we say it's about 300 milliosmolar. That's what's written here. At the top, very dilute, and at the bottom, it's very concentrated. Okay, so remember that concentrated down deep in the medulla, and it's very dilute up here in the cortical area. Okay, and so what's doing that? We can see this other slide over here. We can see that there's something called a sodium-potassium chloride cotransporter. So sodium-potassium chloride cotransporter, and that is actively pushing or pulling, transporting those salts out of the nephron, but only here on the ascending limb. Okay, so So this is another way of looking at it. Now the vasorect is on the other side. It actually goes all around it. So either way you draw it, it's the same concept. The water moves out and then is swept up. The salts move out and are swept down. Okay. Um, so the question is, what happens if we block this? What if we? So here's our co-transporter right here. Okay, so that's moving the sodium, potassium, and two chlorides, moving out all the time. What if we block that? And we just say, okay, salts, you can't move out. And drugs that are called loop diuretics, and the one that's most commonly referred to is called Lasix. These are loop because this is the loop of Henle. They kind of block this. They don't shut it off, of course, but they reduce its function. And so they don't let these salts out. So that means these salts stay in. Okay, so whereas they normally would have moved out, the salts stay in, which means that this concentration isn't going to get nearly as high, for one thing. But it does something else. Here are all these salts in here. Well, the thing that we haven't really talked about yet, because we haven't moved into the distal tubule, is that even though water wasn't allowed to move in the ascending limb, it starts being allowed to move out again. Okay, if you have a bunch of salt in here, how badly does that water want to move out? It says, oh, you know what? Look at all these salts in here. I'm just going to stay in here. Okay? Which is going to keep that fluid in the nephron, which is going to have a higher urine output. Okay, so that's how these guys work. Now, when everything's working just fine, or when you're not blocking this, these salts are gone, and so there's not as much in here to keep the water in. Now, that doesn't mean that water doesn't still want to leave as it moves down and it gets into this higher concentration. Yeah, that water's going to want to leave. Okay, so what happens in the distal tubule and the collecting duct is that our bodies will vary that, and it can say, okay, you know, this stuff all happened automatically down here in the proximal tubule and the loop. That stuff just sort of happens. But what if I want to get rid of more sodium? Okay, what if this person has really low blood pressure? They have very low sodium. We say, okay, well, you know what? We have the power here to put in a sodium potassium pump, and we can draw more sodium out even than was drawn out to begin with. So that means we can pull more sodium out, and then chloride can follow sodium. There's a co-transporter that pulls the sodium out as well. Well, then the water is going to want to leave. Okay, so the water is going to want to leave. Okay, so we just sort of know that. So now what we've done is we've said, okay, wow, our bodies have the ability to actually increase our blood pressure, because that's what would be happening. All it has to do is say, hey, put in more sodium potassium pumps here. Put in more sodium potassium pumps, more salt moves out, and more water moves out. Okay, it has another, and so if we move over here to the text here, it says sodium can be actively transported out of tubule cells into the interstitial fluid just by putting more of these pumps in, and there is a mechanism that does that, or it's a hormone that does that. It's called aldosterone. So when your adrenal gland, that's where it's released, releases aldosterone, it says, hey, put more pumps in there, more sodium moves out, and therefore more water moves out. So sodium, chloride, and water all move back out. Your blood pressure goes up. Okay, and there's another mechanism or another hormone that's released called vasopressin. It's also called antidiuretic hormone here. Okay, so that's what this is, vasopressin antidiuretic hormone. Well, it doesn't do anything with sodium, but there are these little things called aquaporins, water pores, that it can insert, and that's what its job is. Remember that. The ADH, or vasopressin, inserts water pores. Here's the other thing you need to remember. These water pores let water out of the nephron. Okay, so they let water out of the nephron, and people forget that. They forget which direction. They're like, fine, okay, water pores allows water to go through, but you have to remember which direction it's letting it go. When you put those water pores in there, they're called aquaporins. I think I mentioned that. But when you put these aquaporins in here, they're letting the water go back out, which means it's back in the body, which means that it's going to increase our blood pressure, right? Because more fluid in our body means more pressure. Okay, so we might have a drug that um, either blocks that or increases activity of that. Alcohol blocks these, so you don't have as many aquaporins, which is why we tend to urinate more. Okay, so by putting the water pores in, instead of becoming urine, they leave. If we don't have as many of these water pores, these aquaporins, then that fluid will have to leave as urine. Okay, so what does that do for the concentration of the urine? Well, if you let sodium and water out, okay, you're going to, or the volume of urine. So if you let sodium and water out, that's going to reduce your volume of urine. If without having much of an effect on concentration. However, if you leave those salts in there, so we have a whole bunch of salts in the nephron here, and then we put water pores in where the water can just leave, then what we end up with at the end, and that water is going to leave because look, it's 1200 milliosmolar. It's very, very concentrated, and water is going to go to where it's very concentrated. And so as long as the concentration is different, it's moving out. So as that water moves out, it's leaving those salts behind, and we end up with a very highly concentrated urine. Okay, so it's going to roughly be the same as it is down deep in the medulla. If we don't let the water out, okay, and let's say we're pulling salts out still up here in the distal tubule, but if we don't allow the water to leave as well, then all of that water stays in the urine, and we have very dilute urine. And we've all seen it. You know, you see the different colors of yellow when you're assessing someone. And you look at their urine, you can say, okay, well, this urine is very clear. There's hardly any yellow color to it at all. So that must mean they're extremely well hydrated. Hopefully it's not because of some kind of pathology, but they're extremely well hydrated. Or you may see that it's a very, very deep, deep amber, dark yellow, and you say, wow, this person is very dehydrated. Okay, so they don't have very much water in their body at all. Okay, so that means that we can vary that urine concentration, and those are two big ways that that's done. All right. So urea, we haven't talked about urea, but we hear about it a lot. So urea is just when these proteins, we have proteins that do all kinds of stuff in our body, but they're broken down into their individual amino acids. Now, amino acids tend to really change the pH, and so urea is a molecule that's made from the breakdown of these proteins into their amino acids. That's a little more neutral, and urea is really a harmless molecule. It doesn't do anything. It's not toxic, so it doesn't, you know, within reason, too much of anything is bad. But urea is just kind of floating around in our bloodstream all the time. Well, so when filtration happens, it ends up in the filtrate too, and it moves through. But there's kind of a trick with urea, and what urea tends to do is it moves out of the collecting duct, and then will be reabsorbed in the loop of Henle. But it kind of keeps it low down in the medulla, mostly because of the concentration that's in there. But by doing that, the urea will add to, so remember, the idea here is that we want a high concentration deep in the medulla. And so the urea will actually contribute to that as well. So it's kind of constantly circling around here. About half of it will leave, so about 50% of it will actually leave as urine, but we've got a pretty constant supply all the time, and so it's kind of looping, cycling around in this area, which is contributing to that concentration gradient, as well as the other salts that we talked about. So descending limb and descending limbs of the loop of Henle are permeable, so it can move back in there, and then it loops around. So it flows in that cycle, and that's going to maintain the high urea concentration in the medulla and contribute to that concentration gradient down there. Okay, so now the we've already kind of talked about how when blood pressure is really high, and we've talked about the control of GFR. So when our blood pressure is high out here, don't want things to just go shooting and through the nephron. We want to maintain that, because if we have really high pressure, then we're going to get way too rapid of flow. Now, your nephrons themselves are constantly monitoring this flow. And they do that because even though we've been sort of looking at our picture like this, we've been looking at our nephron as being filtration happens here, and then it kind of moves in this direction. We've kind of been doing it in a left to right fashion, but that's really not the way it is. So what happens is that as filtrate moves through the proximal, then it goes down the loop and then back up, and the distal goes right past these arterioles. Okay, so you kind of see that right there. The distal tubule is in direct contact with these tubules. What that means is that there are a bunch of cells here they're called the macula densa, because I guess that's what they look like. So there's this group of cells that are right there among all of this, and they are monitoring the flow through this distal tubule. And they may say, okay, we've got too much sodium, or we don't have enough sodium. And because they're right here with the afferent and efferent arterioles, they can say, okay, we have too much sodium, we need to slow down our filtration rate. So they'll activate those smooth muscles, and they'll constrict this a little bit more, and they're going to slow the filtration. So this filtration is going to happen much more slowly. And what that's going to do is it's going to allow more sodium to be reabsorbed. Okay, so what we call this is the juxtaglomerular apparatus. It's kind of a cool name. So the juxtaglomerular apparatus it sits right here, and that's that loop back on itself. And so we can see this better in this picture. And so that's kind of this is where it's blown up, and you can see here's the afferent arteriole, this is the efferent arteriole, and this is the juxtaglomerular apparatus, or these are the juxtaglomerular cells, which is called the macula densa. I know all these words, but that's what's happening. Is this is our distal tubule, this guy right here, and so these cells are constantly monitoring how much sodium is in there, because that sodium is going to move across, and when more sodium is there, it activates it, and it says, okay, too much sodium. Contract these smooth muscles and slow this thing down. So it also looks at pressure, fluid pressure that's in there, and will react the same way. And so it's actually monitoring what's moving through there. Now, one of the other things that this the JG, it's easier to say JG than juxtaglomerular. So another thing that it does is that there are cells in there that release a molecule called renin. Now remember, I wrote RAA for renin, angiotensin, aldosterone. So we're going to come back and we're going to talk about that. So remember, this is where this monitoring is also where renin, the hormone renin, is released. Okay, so more on the adjustments of GFR. Autoregulation, we've already mentioned really these. As systemic blood pressure increases, the afferent arterioles constrict, okay, which is going to slow movement into the glomerulus, which is going to slow the rate of filtration, your GFR, glomerular filtration rate. So, and then we just talked about as the rate of blood flow, the filtrate past these cells of the macula densa. If that increases, they're going to constrict. They're going to say, okay, slow down, slow down. We've got too much in here. Now, there's another thing that we haven't really talked about, but kind of makes sense, and that's sympathetic. Remember, sympathetic is your fight or flight. That's your adrenaline rush. Well, adrenaline, epinephrine, norepinephrine is the neurotransmitter that controls that. Okay, and what that does is it causes constriction of the small arteries of the afferent arterioles. So sympathetic nervous system activation. So if something frightens you, or during exercise, that's going to lead to constriction. Okay, during shock. Okay, so this during shock is that's going to cause a release of norepinephrine too. So shock is when your blood pressure falls to the point that you actually can't you can't feed your cells enough. Your blood pressure is so low that you're just not even able to circulate fluid through to get to your cells. So what that does is it shunts it away, and it says, okay, we're going to reduce blood flow through the kidneys, and we're going to keep that in the systemic circulation. The rest of the body is going to be able to have that. Okay, so let's go back and talk about this renin angiotensin aldosterone mechanism, the RAA. So we talked about this in the circulatory system. So low blood pressure, so when your blood pressure really starts to fall, that's going to cause the kidneys to release this molecule renin. And remember, that was at the juxtaglomerular apparatus, okay, those macula densa cells. So the JG cells will release this molecule called renin. And I have these in red because those are the big things that happen. But that renin is going to cause this molecule called angiotensinogen, which is made in the liver, to it's going to break that down or cleave that, cut it, and form something called angiotensin 1, and then angiotensin converting enzyme, which is made in the lungs, will convert angiotensin 1 to angiotensin 2, and angiotensin 2 is the one you really need to remember. Okay, that's why it's in red right here. Angiotensin 2 is a potent vasoconstrictor. Overall, even though some things may be different in terms of vasoconstriction, vasodilation, but overall, when you have vasoconstriction that takes place, that's going to increase blood pressure. Okay? Massive body-wide vasoconstriction is going to increase blood pressure. Well, that's nice because this whole process started out because you had low blood pressure. So now you have vasoconstriction caused by angiotensin 2 that's going to increase the blood pressure. Another thing that it does, and this is what's important for the renal system, angiotensin 2 stimulates the adrenal cortex to make a hormone called aldosterone, okay, which is the third part of the RAA. The renin was released by the kidney, converted, we ended up making angiotensin 2, and the last A is aldosterone. Aldosterone, now if you're paying attention to the previous area in the distal tubule, aldosterone increases the number of sodium-potassium pumps. So that is going to, this is what you really need to know. Aldosterone increases sodium reabsorption, okay, which is going to keep sodium in your body. If you have more sodium in your body, the water is going to stay in your body. That's also going to increase blood pressure. Okay, sodium stays in, then chloride is going to be co-transported back in, and water is going to either remain or follow it. So as a result, your urine volume is decreasing because you just pulled all of that water. You pulled the sodium out. That's what aldosterone does. It causes sodium to be reabsorbed, and then the water is going to be reabsorbed. So none of that is staying in the tubule where it used to be. So it's in the body. Your blood pressure increases. Your urine volume decreases. Okay, so that's and then this is a cartoon you can look at, whatever. I don't know. I don't really understand it. It's kind of hard to read, but I get it. And it says all the things that we have to know, so that's why I put it in there. But here's the problem. Drugs that affect the RAA system can affect potassium. Why? Because it's being kicked out by the sodium potassium pump. And remember, the sodium potassium pump, yeah, it pumps out 3 sodiums to 2 potassium, and it burns an ATP in the process. ATP, misspelled ATP. But the thing is, as we're pushing the sodium to go back into the body, this potassium is being put in the nephron. And because it's being put into the nephron, it ends up leaving the body as urine. Okay, so drugs that affect the RAA system can affect potassium levels, so the patient needs to be monitored, and that's because it's a co-transporter. Yes, the sodium's going back into the body, but the potassium is leaving the body. Okay, So this kind of goes through the whole thing. So renin is released at the juxtaglomerular apparatus. So we talked about what that was, which causes, which ultimately, we skipped a few steps here, but ultimately causes the release of aldosterone. So now we're talking about what aldosterone does. Aldosterone inserts more of these sodium potassium pumps into the distal tubule and the collecting duct, and it's really the first part of the collecting duct. So it's mainly the distal tubule and the first part of the collecting duct. So reabsorbing more sodium and the chloride that follows it is going to lead to more sodium retained in the body. Well, this is why we avoid sodium, because sodium in the body is going to increase that osmotic pressure, which means that we need more water to offset that extra sodium, which is going to lead to higher blood pressure. Okay, so aldosterone activation of the RAA system leads to increased blood pressure. Okay, so aldosterone lowers urine concentration and volume, and because so many organ systems are involved, remember we were talking about the liver, the lungs, the kidney, because so many organ systems are involved, any kind of diseases, liver disease, lung disease, kidney disease, can severely impact your ability to control your blood pressure, because this is a major way that the body controls its own blood pressure. Okay, vasopressin. I kind of mentioned vasopressin, and that's the one that inserts the water pores. So if we remember the vasopressin, the release of vasopressin from the posterior pituitary. Remember, only two hormones are released from the posterior pituitary: vasopressin and oxytocin. So when vasopressin is released, it inserts these water pores and allows water to move out. That's the real thing you have to remember. I know I mentioned that before, but that's really important. Um, so vasopressin is going to allow more water to move back into the body, which means that you have more fluid in your body, which is going to increase your blood pressure. So the RAA system and the vasopressin system both are going to increase your blood pressure. Now, something like alcohol blocks vasopressin and will leave that water in the tubule, which means that you're going to make a whole lot of very dilute urine. Okay, so that's why you pee more when you have alcohol, because it's blocking this mechanism right here, the vasopressin. Okay, and it works as a diuretic. Okay, so diuretic is just something that causes you to increase your urine volume. Okay, so here's the question. I've already kind of answered it. If vasopressin, which is released by the posterior pituitary, increases reabsorption of water back into the body, what you have to remember, what effect would that have on blood pressure? Well, hopefully you say, well, if it's going back into the body, it's going to increase blood pressure. And that's exactly right. What does it have? What effect does it have on urine output and concentration? Well, it's going to make very, very concentrated urine, because there's no water. The water is going back into the body, which means it's not staying in the urine. So it's going to reduce the output, the volume of urine, and it's going to make it very, very concentrated. So when you're dehydrated, vasopressin is going to be released. Okay, because that it's saving water. It's keeping the water in the body. Okay. So a summary of urine volumes and concentrations. So the countercurrent mechanism, we've gone over that several times. This opposite flow, water comes out on the left, salts come out on the right, or the descending water comes out, ascending sodium chloride and potassium come out. And then you have the counterflow of the vasorector, or that capillary, that's sweeping the salts down and pushing the hydrogen or the water up. Okay, and so that's going to cause this concentration gradient right here. So from 300, which is pretty dilute, to 1200, which is very concentrated. Aldosterone increases sodium reabsorption, so we remember that, and that's part of the RAA pathway, renin-angiotensin-aldosterone. ADH, or vasopressin, both words, it's important to remember both words, causes aquaporins to be inserted, and we remember that those aquaporins are allowing water back into the body. So that's going to increase blood pressure. And also GFR, I don't know how much I've mentioned this, but if we increase the rate of filtration, that's also going to lead to more urine production. Okay, because the more that moves into the nephrons, the harder it is to reabsorb all of that, and so we end up with more volume. I hope that makes sense, that by letting more fluid flow in, so allowing more to be filtered, means that you're going to have more that comes out as urine. It's doing the best it can to reabsorb as much as it can, but that's why we want to keep our GFR pretty constant, because if we let it go, it's just going to flow through there, and that's so increasing GFR will decrease blood pressure. So if we let more fluid go, we're going to release it as urine. It's going to move through those nephrons, and so that's going to decrease blood pressure. So if you increase your urine volume, you're decreasing your blood pressure, and you have to keep those things straight. And it kind of makes sense, because more urine, there's more urine, that's water that's not in your body anymore. So it's going to have a direct effect on blood pressure. Okay, so overall, the kidneys keep hydration at safe levels. When we drink a lot of water, we're able to get rid of it. So we can eliminate excess without losing large amounts of electrolytes. So we have control over that. We could say, okay, we need to keep our sodium, but we don't need all this water. We have way too much water. This person is sweating a lot, but they're just drinking regular water, and so we just need to get rid of the water, but we need to maintain as much salt as we can. So our body's able to do these things to a point. It can't forever if you don't replenish your salts, but it can within a range. Okay, and when drinking water, so when we're very dehydrated, we're not drinking anything, which could be worse, then it's able to reabsorb both the water and the salts, or whatever it has to do. Okay. All right. So one thing that we haven't talked about is secretion. Okay. So if you have, so we know what filtration is, so things are moving through filtration. So let's say we have 100 per minute, I don't know, whatever, milliliters, milligrams, whatever. It doesn't really matter. But if we have 100 coming through and we lose, let's say, 50 through reabsorption, so we have 100 milliequivalents of glucose. Well, glucose is a bad example. Potassium. So and 50 is reabsorbed. Well, we have the possibility, so that's only leaving 50 milliequivalents there, but there's this little possibility of secretion, of bringing it back from the blood, and that's what secretion is, from the blood, and putting, let's say we put in 20 more. That means we're going to excrete 70. Okay. So do you kind of see how that works? It comes down to this little equation. However much is filtered, the amount that's filtered minus what's reabsorbed. So this is what's in the nephron and what's going to be urine. So the amount filtered minus what's reabsorbed plus whatever gets added back by secretion. And that's going to be the amount that's excreted. Now here's a great example, and this is penicillin, because the thing about penicillin is that it's really not reabsorbed at all. Okay, so there's no reabsorption of penicillin, but there is secretion. So here we can see exactly that. Filtration, 100 milliliters per minute. So there's some good units for you. 100 milliliters per minute are filtered. But now let's look down. Let's skip all this stuff in between, and let's look down here, and we're saying, oh, we have excreted 100. We're excreting 150 milliliters per minute. Maybe you've already figured this out, but the question is, how on earth are we doing this? How are we excreting coming out of the nephron? How is more penicillin coming out of the nephron than went into it through filtration? And of course, we already know the answer is that as this penicillin moves through, 100 milliliters per minute, some of it escapes by, but when it gets around here, it says, nope, you're going into. So this penicillin thinks it got away and wasn't going to be excreted, but then there's a pump here that says, nope, you're going into. So it adds it to it. So here's the 100, but there's the other 50. It's moving right in, right through here. And that's what secretion does. Okay, so that's secretion movement from the peritubular capillaries into the nephron tubules. And it does. It moves byproducts, drugs, molecules not normally produced by the body, and there are certain pumps and channels and stuff that are specific to certain molecule types that will secrete those and move them in. Now, the ones we're going to talk about next are hydrogen and potassium. Hydrogen, of course, is important for pH, and potassium, we want to maintain our potassium levels. So they're actively secreted into the nephron too, and we can see that here. Kind of, these pictures are kind of small, but we can see that there's some counter exchange. So if we look down in this area, so this is our nephron here. Okay, this is the nephron tubule here, or filtrate. And we can see that as the sodium leaves, hydrogen can be pumped or moved, counter exchange. It's a trade. So hydrogen can move down into the filtrate. Well, that's a great way to control blood pH. So if we become too acidic, acidosis, then we have a mechanism. It's fantastic. We can just boot that hydrogen right out, and that's a secretion mechanism. Okay. Now, also potassium. Potassium can also be secreted, and except, let's see, we're gonna come back and talk about this again when we talk about ions, but I have here, and this is something to remember, potassium is only secreted in the distal tubule, while hydrogen is in the distal tubule or the proximal tubule. So remember our distal tubule. Distal tubule is over here. We know that's not really the way it is, but that's easier to understand. And here's our proximal tubule here. So hydrogen can be secreted. That now we're going from the blood back into the nephron. So hydrogen here and in the distal. Potassium is just in the distal. Okay, and it's this counter transport mechanism. Okay, so that's just something you need to remember for now, but we'll hit it again a little bit later on. Okay, so urine production summary. So how to make great urine. In the proximal convoluted tubules, most reabsorption takes place. We'll remember that, won't we? Sodium, sounds so condescending. We'll remember that, won't we? Yeah, we will. Okay, so proximal, most reabsorption takes place, and remember this. I don't know how many times I've said it, but sodium is actively removed. When I say actively, I mean that it's burning ATP. That's what I mean. That means that it's using energy to do it. That's what active means in this case. So sodium is actively removed, but then other things are removed with that. So they use that sodium concentration gradient and other things like chloride, amino acids, glucose, other ions are being removed. Okay, so all of these other things are following it. In the descending loop of Henle, we remember that too, hopefully, that water exits. Okay, water exits, says passively, it's through osmosis, but water, it's permeable to water, so water moves out in the descending loop. In the ascending loop, the sodium potassium chloride cotransporter, that's also active, so it's using the sodium potassium pump to boot those guys out. Okay, that's in the ascending loop or limb of the loop of Henle, and then the distal, you have these other mechanisms. Antidiuretic hormone, which is vasopressin, okay, and then aldosterone. So vasopressin, remember that's the one with the aquaporins, okay, or water pores. That's vasopressin, which lets the water out back into the body. Aldosterone, remember aldosterone is mainly reabsorption of sodium, and then chloride will follow that, and then water will follow that. So, but we remember aldosterone, we associate that with sodium. Okay, well there's another hormone too, and that's called atrial natriuretic peptide. So atria, and in this case we're talking about the heart. Okay, natrium, that's going to refer to sodium, and uretic is going to refer to urine. Okay, and it's a hormone, so it's released into the blood. So atrial natriuretic hormone. So what happens is we have the heart up here, and in the right atrium may stretch because you've got too much fluid in there. Okay, so too much blood, and so your heart releases this peptide. It's a peptide. That's what oh, I didn't notice that. It's ANP. P stands for peptide, but it is a hormone. Um, it's just a string of amino acids. So I didn't mean to make that confusing. But the atrial natriuretic hormone, or atrial natriuretic peptide, is produced by the right atrium of the heart when the blood volume increases and stretches the cell. So it goes down and it says, okay, aldosterone, or whatever, has been telling you to reabsorb more sodium. ANP, atrial natriuretic hormones, say stop reabsorbing all of this sodium. Let the sodium leave. We've got too much blood. I'm stretching. Okay, so this sodium is no longer reabsorbed. Well, it reduces the reabsorption, and that sodium then leaves in the urine. Okay, which is going to, in this case, decrease blood pressure. Remember, the RAA system and the vasopressin increase blood pressure. This is the only one of the three we're talking about anyway that decreases blood pressure. Remember that. Okay, and it also inhibits the ADH, which is going to leave the water. So if it inhibits ADH, that means that ADH is vasopressin. It's going to leave that, leave the water, and you have a whole lot of urine. You increase your urine production, and your fluid leaves your body. Okay, so it increases the volume of urine produced, and therefore decreases blood pressure. Okay, so now we're going to move, we're almost done here, but we're going to move from the kidneys, we're going to move through the ureter. There's a peristalsis that takes place where you're actually actively trying to move things. So peristalsis, remember, that's that wave-like function that pushes things forward. So as the urine is being produced and dripping into this renal pelvis, the calyx in the renal pelvis, then the ureter will push it down into the bladder. Okay, and the bladder will expand, and then at some point you release that as urine through the urethra. So a couple of things. First of all, the male urethra goes through the penis. So from the bladder through the penis, and the female is going to be shorter, okay, because there's no penis, and opens into the vestibule anterior to the vaginal opening, so near the vaginal opening. And because it is shorter, and that's what's important here, is that it's more susceptible to urinary tract infections. And of course, if you have a urinary tract infection, that can, and you don't control that, it can lead to a bladder infection. And if you don't control that, or if it's just a really nasty one, it can move up through the ureter and become a kidney infection, and that's not a good situation. Okay, so the last thing, very, very quickly here. So when this urine that's been produced by the kidney moves through the ureter, and the ureter pushes it down into the bladder will stretch, and there are mechanical receptors on the bladder that say, wow, we're stretching a whole lot, and so it will send a signal back to the spinal cord. So this is a reflex type of signal, and it sends a signal saying, hey, we're stretched, and it's time to go. And it's going to send a signal back. This is a reflex. Sends a signal back to the bladder telling the detrusor muscle, which is the name of the muscle that's around the bladder. It says contract, and that will squeeze the bladder. However, we know that isn't always what happens. Sometimes our bladder stretches, and it will then, it also sends a signal up to the brain and says, hey, we're stretching and we're about to pee. And the brain says, yeah, no, that's not really a good time. So it can send a signal back to override that signal, and you can actively, consciously constrict what's called the external urethral sphincter. And it will pinch it, and it will close it, and it will prevent it from opening. Now, when you're ready to go, you can relax this external urethral sphincter, and that will then allow this reflex to take place. Okay, and then the detrusor muscle will contract, and it will push the urine out. Okay, so a lot of problems can take place in there. I don't know if we haven't talked about it yet, but the prostate, the urethra in the male goes right through the prostate. So, sure, we don't have to worry as much about urinary infections, but, we've got that little issue. Um, and then any kind of sometimes of spinal injury or spinal nerve or neuronal injury, neurogenic diseases, whatever, can affect with this and lead to incontinence where we're not able to control it. Okay? All right. Well, that's it for chapter 26, whatever we're in. And I'm sorry this was so long.